So I just want to asking the report also include the um, annotation of the mutation, like ACMG report for somatic mutation. Like last week we have the presentation from the Qiagen. They
Hmm.
have a database for
Yeah.
annotate on the somatic mutation. So how about this report? Do we include the annotation database as well?
So for right now, our report doesn't include any annotation yet. So we only generate the reports out to actually help you to identify all the different, mutations that you have, but we don't annotate it. But I understand like from Kaizen, they have like, QCI and that can actually help to annotate with like Cosmic as well. So it's very, I mean, yeah, so we can actually generate VCF file from our WDL pipelines, and then we can also work together with Kaizen. Let's say you need annotation, right? So they can actually import our VCF file in, and then generate those annotation from their software. Mm. But
Oh,
right now for PacBio, we don't offer our own annotation yet. But I think our computational biologist team is actually working on that as well. Yeah, but it's not ready yet. Yeah.
okay, okay. Thanks a lot. Thanks a lot.
Thank you, Dr. Loy, for your question. Okay.
Just to answer one question before you move to the next section.
Yeah.
Um, for the sequencing RNA-seq,
Hmm.
can we detect on the RNA modification like the SIG-MA, some modification like DNA methylation, but in the RNA?
Yeah, so I think the we can so there are customer developed protocol that they did some pre-treatment before sequencing it through our PacBio sequencing. So we do have some publication where some customers actually develop some pre-treatment protocol. So they change some of this like M6A modification to an alternative base, and then after that they sequence it on PacBio itself. So by doing that, we can actually detect DNA modification. Yeah. So compared to our competitor, I think Dr. Loy, you want to, what our competitor's strength is, of course, they say they sequence through like native RNA, right? So that actually enable them to actually look at the modification part. But I think it still depends on the application of what you want to look at. If let's say the modification that I think they are generating is also just a probability right now, like they just tell you maybe 60% this particular base has a modification, but it's not like 100% sure yet. And then I think our strength is also in terms of let's say you want to look at RNA variant calling. So our bases are of course much more accurate compared to what they can actually offer.
Yeah, thanks a lot. And how about the kinetics with the RNA-seq from the hi-fi backbio sequencing? Can we it will adapt for all the other uh, single cell platform like the 10x genomic or the other platform as well, or only specific for some of the platform for the single cell technology?
Yeah, so our Kinect single cell platform, I mean, the Kinect single cell kit is optimized on the 10x platform. And then there's also a new protocol that came out for Parse as well. So if you're using Parse, it's also compatible with our Kinect single cell workflow. Hmm.
Okay, it means that we don't need to convert anything, just directly use with the power.
Hmm.
Okay.
Correct. There's a
Okay.
protocol that we actually have that you can actually use directly. For,
Okay. Okay. Thanks a lot.
okay. So I'll go to the next section.
Hello.
Hi.
Yes. Thank you, Ryland, for your express brief video express now. I want to ask question for the slide in ACA 1A. I don't remember at what do you want to share?
Which one? Sorry. Do you remember
Ờ,
which
ờ,
slide?
Doctor lại lưu send like to me. Um, cái phương pháp mà IT A1A là cái phương pháp đó như thế nào, có thể chia sẻ lại.
Cái slide số mấy á, anh nhớ không ạ?
Anh không thể nhớ, nhưng mà cái
Anh gửi em cái tên đi để em dịch lại.
uh, ok.
I'm sorry, like
A moment, I'm trying to translate his
okay,
question
okay.
to you. Okay. Anh gõ tên cái công nghệ đó vô đi rồi em hỏi anh Ray cho. TD1A.
Xin lỗi anh, em cũng không biết công nghệ đó là cái gì với anh. Xin lỗi. Dạ.
Trong slide luôn hay sao anh? Hồi nãy em cũng không thấy nữa.
One slide in the middle online on slide.
Alright.
Ray,
can you re-explain about like?
Yeah.
Can you please re-explain about the technology DTD1A? I don't know which slide, but he want you to re-explain about the technology name DTD1A. He typed in the Zoom chat box. Can you see it?
The TD1A.
Yes, I'm not sure. Where is it?
Do I remember? Is there a
Ở trong slide luôn đúng không anh ơi? Nãy
yeah,
hình như
in on slide.
about mà về cái
Before TF DNA ctDNA before slide.
before slide?
Yeah.
please return until he's
Trying to find where the one is.
anh đang move lại đó, cái đến slide đó thì anh bảo dần nha anh, em cũng không nhớ nằm đâu. Here. Okay.
Here.
Next slide.
Next slide.
Okay, this one. Sorry.
ID1A.
Yeah, your question is
He not really understand about like the whole slide here. So can you please to re-explain again for him?
oh, okay. So I think the main message from this particular slide is so they were actually looking at gastric cancer, right? So they actually use our long read isoform sequencing to actually look into this particular gastric cancer. And then they found two new isoforms for this particular gene, this AR1D1A gene. So they found that there was two new particular isoform, and then this particular isoform, right? One of them actually have a truncated, actually uses a new novel promoter. So this particular novel promoter actually resulted in a truncated protein. So this particular novel isoform, right, actually, truncate the protein through the usage of a particular alternative promoter. So if you are using short read sequencing, right, you won't be able to actually detect this particular novel isoform itself. But by long read sequencing because we sequence through the entire mRNA. So that's why you are able to actually, detect these two different isoforms for this particular gene. And then they were actually looking at this particular, I think this is um, what we call the Kaplan-Meier curve, right? That actually shows, I think, the survival of a particular patient itself. And then you can see that for this particular novel promoter, it actually the survival rate for the particular patient actually decreased by a lot. So the number of days actually, are shorter compared to this particular. If the particular patient has the isoform with a known promoter, they can actually survive longer. But if let's say they have an isoform with this novel promoter, it's a biomarker for actually a worse survival. So means the patient particularly will die earlier because they have this particular isoform with this novel promoter. So which means that by being able to look at the different types of isoforms, we are able to actually identify this kind of biomarkers that will actually help to predict predict the prognosis, or tell the prognosis for a particular patient itself. So by having this technology itself, pharma companies, they can also look into this kind of biomarker, right, to see whether, can they, develop a particular drugs to actually target this particular isoform, so that it can actually help to, prolong the life of patient itself. So yeah, so I think this particular study is one, just want to say, because of them using a long read sequencing itself, they are able to see new isoforms that can actually, predict prognosis of a particular patient.
Dạ anh ơi, anh ok chưa?
Ok,
Dạ,
I believe that.
tức là tại vì em giải thích ngắn ngắn lại xíu ha, tại vì anh nói tức là cái long read này nó giúp mà phát hiện thêm những cái isoform mới, tại vì nó giải từ đầu 3 phẩy đến 5 phẩy thay vì short read thì nó không không có hết, nó chỉ nhìn được một phần của những cái transcriptome này thôi, còn cái này nó phát hiện hết, cho nên là người ta mới phát triển thành biomarker mới để phát triển thuốc mới, thì cái đại ý là vậy ạ.
Vâng, cho anh nói tiếng Việt em dịch giúp đó. Tức là
Dạ,
tại vì
dạ.
cái này nó là một cái mặt cơ lạ nên anh quá chú ý, nhưng mà nói chung là cũng sẽ có được nhiều cái mở rộng cái hiểu biết hơn, nhưng mà thực sự là nếu ở Việt Nam thì hình như chưa có làm được cái mặt cơ này đâu.
Cái đó chắc là anh Ray sợ anh không giải thích được anh, cái này anh chỉ nói về mặt công nghệ thôi, còn những cái ứng dụng của cái này như thế nào thì sẽ là những cái nhà nghiên cứu hoặc là những bác sĩ anh, còn như cái ý mà anh muốn nói ở đây tức là
Không,
nó
ý anh muốn nói là
ừ.
hiện tại ở Việt Nam chưa có cái công ty nào nó làm cái mặt cơ này á, nên anh chú ý gọi là anh nhìn thấy cái giới thiệu cái mặt cơ hơi lạ nên anh muốn cho mình rõ hơn. Rồi anh
Thế
sẽ ghi nhớ là lần sau anh sẽ có cần thì anh sẽ đòi hỏi. Rồi cảm ơn.
dạ cảm ơn. He just asking, do you know any Vietnamese company that already developed the biomarker that you mentioned in the slides?
I'm
Do
not
you know?
very sure.
Anh không biết anh không chắc rồi bác ơi, chắc là anh chỉ nói về mặt công nghệ thôi, còn đương nhiên là chắc chắn sẽ có, nhưng mà Việt Nam có không thì anh không biết. Dạ em cảm ơn ạ. Thank you, Mr. Ray.
No
You can,
problem. But I think like the question is valid lah. I mean we know this kind of technology is new, right? And then there are many cancer type that are maybe unique to the Vietnamese population itself. So like what I mentioned, right? Like in the earlier on, I shown a few of the study itself that for things like even breast cancer, that was very well studied, right? But it was well studied on short read itself. But if we can apply long read sequencing to actually do both genomic DNA sequencing as well as transcriptomic DNA sequencing, there will be many, new discovery or many new things that you can actually, I mean, to be able, just looking back at any of the cancer samples that you have, if you do long read sequencing on it, you will be able to detect a lot of the new things that you might not be able to actually see in short read sequencing itself. Yeah.
Thanks a lot for your information. Dạ,
Hmm.
dạ ý anh nói đây là cái này giống như là long read thì sẽ giúp có nhiều thông tin hơn, có thể là sử dụng những cái mẫu cũ trước đây mình bằng short read, mình giải short read mình không có biết được những cái thông tin quý giá đó, bây giờ mình dùng long read thì mình khám phá thêm nhiều thông tin hơn đó anh. Thì cũng giống như nãy em nói từ đầu á, còn cái việc mà ứng dụng những thông tin đó, tìm hiểu được đó như thế nào thì là chắc là thuộc về chuyên môn của bác sĩ hoặc là những cái nhà nghiên cứu về ung thư á. Dạ em cảm ơn ạ. Thank you. You can move to the other slides.
Okay. Okay. So I think, should I continue, or if there's anyone that would like to ask any questions?
I received one question in Vietnamese, but it's too long, and I think like let me translate for you, but I'm not sure I translate correctly. Can you see in the chat box? I just summarize his questions. He has 4 questions here. So can you see on the chat box, Mr. Ray?
Yes, I see.
Yeah, one by one, please. Thank you.
Yeah, so I think the first question is like whether the application of PacBio IsoSeq is economically feasible in Vietnam, right? I think for right now, if you do keep track of PacBio sequencing itself, right? So from PacBio sequencing itself, our price of sequencing has been decreasing every year. It's actually decreasing because of the new technology. For example, when we launched the Revio, we can actually run, a 90 GB on one smart cell itself. So we are able to generate genomes at, 30x genome at 1,200 USD at that time. And then after that, we also launched a new chemistry like Spark chemistry itself. So the Spark chemistry also, increases the throughput of the Revio itself, and then, further bringing the sequencing cost down. So I think for PacBio IsoSeq right now, and together for IsoSeq itself, we have the new kit on Kinex itself. So with the Kinex itself, the Kinex kit will actually help to increase the throughput of the isoform. I mean, the throughput of the RNA reads that you actually get from one single smart cell. So because of all this different technology, right? The IsoSeq is getting more and more economically flexible. That's why you see, I think in these 2 recent years, there's so much more isoform related publication that is being published nowadays.
Thank you. Let me translate first. Anh ơi, cái câu đầu tiên là hỏi nó cái IsoSeq nó có hiệu quả kinh tế hay không á, thì anh nói là nếu mình theo dõi PacBio từ lâu á thì mình cũng có thể biết được là giá thành ngày càng giảm dựa trên cái team R&D của họ, họ làm rất là mạnh luôn. Thì ví dụ như là trước đây giải trên cái Revio System á, thì bây giờ nó có những cái hệ thống khác thì nó sẽ làm giảm giá thành nè, thứ nhất. Thứ hai là họ cũng phát triển thêm một cái chemistry, em nhớ là tên gì, nó cũng làm tăng cái throughput của cái tức là cái thông lượng mình giải của cái hệ giải trình tự. Cái thứ ba là nãy anh có nói cái Kinex kit á, tức là ví dụ như là cái giải hi-fi của anh là từ 25 đến 30 KB đi, còn của mình nó ngắn hơn thì mình có thể dùng cái Kinex đó mình concat lại, tức là mình ráp nó lại để mà mình giải được nhiều mẫu hơn, thì cái giá thành nó sẽ giảm xuống rất là nhiều. Thì ý anh muốn nói đây là cái team R&D đang làm, nói chung phát triển rất là nhiều thứ để làm giảm giá thành xuống từ nãy em nhớ không 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 lầm là từ giống như từ 1.000 đô mà xuống còn 100 đô vậy đó, thì nó sẽ rẻ hơn rất nhiều, nhưng với điều kiện là mình phải gom đủ mẫu lại, và đương nhiên là mình cũng phải có cái chiến lược vun mẫu sao cho nó hợp lý á. Thì cái đó em trả lời thêm, còn ý anh nói đây là giá thành đang giảm, nhưng mà nó chỉ rẻ khi với điều kiện mình giải được nhiều mẫu cùng một lúc. Ví dụ mình có cái chiến lược gom đủ mẫu trong cùng một thời điểm hay gì đó, mình giải thì nó sẽ rẻ hơn rất là nhiều ạ. Dạ em cảm ơn ạ. Okay, please, the next questions, please.
The next question is like, I think he's asking about whether, have we done IsoSeq on fresh or FFP tissue samples itself, right? So I think the crucial questions about RNA It's always RNA is very sensitive, right? So we always, I mean, on the FFPE samples, I think even on the short read side as well, we know that RNA is very sensitive. You know, if you go through like FFPE process and everything, the RNA will be degraded itself. So I think there's not a lot of studies for isoseq on FFPE. There are more studies for isoseq on fresh frozen tissue or like blood samples. Uh, but I think for FFP tissue samples, right, I will talk about a new technology which is on my screen right now, which is called Amplify. So on the DNA level, you know, we are actually enabling some of these long read sequencing for FFP tissue samples, which is very exciting.
Thank you, let me translate. Anh nói là cho cái mẫu FFPE hay là mẫu tươi mà anh nói á, thì tại vì mọi người cũng biết là cái RNA nó rất là nhạy cảm, cho nên là cũng không có quá nhiều nghiên cứu mà về cái FFPE tissue này, mà nhiều hơn là cho mẫu máu hay là cái mẫu khác. Nhưng mà cái FFPE này thì anh sẽ đề cập đến cái công nghệ mới mà anh sắp nói nha, thì nó sẽ giải cái mẫu FFPE nhiều hơn ạ. Thank you. The next one, please.
Okay. So I think the third one is about whether are we developing therapies based on specific RNA isoforms, right? I think there are like a lot of different applications. I think a lot of the different publications that, you know, why they want to, you know, look into new isoforms or like new biomarkers. A lot of times, you know, their final goal is actually to develop, you know, a ASO or like, you know, a vaccine kind of thing for, you know, downstream work, whether is it on the rare disease side or the cancer side itself. So there are ongoing efforts. But whether, you know, as we know, all these therapies need to go through clinical trial and things like that. So it will probably take a few more years before they are able to generate out.
Thank you. Dạ cái câu này thì cũng anh cũng nói là đương nhiên là có rồi ạ. Okay, thank you. The last one, please.
Yeah, so I think the fourth one has Vietnam plan to build a cancer isoform atlas. I would be happy to support if there's you know a research team that is, you know, looking into building a cancer isoform atlas in Vietnam itself. So we know what I know is like the Japan team, I mean the Japan on the Japan side, they do have this cancer genome atlas program that they are also, you know, using our long read sequencing to actually support that program. I think in the Singapore side, they have this like they have human cell atlas that is, you know, trying to profile every single cell's expression. I mean, in every cells like in healthy patients, I think, but they are also, you know, exploring long read sequencing right now to actually, you know, go into isoform level information.
Dạ, thank you. Dạ cái này anh nói là hiện tại thì ở Nhật Bản với lại ở Nhật Bản thì có cái Cancer Genome Atlas. Dự án này thì là bên ảnh cũng đang hỗ trợ ạ. Còn Việt Nam thì nếu mà có thì ảnh cũng rất là vui để mà, đương nhiên là Parkwell rất là vui để mà hỗ trợ. Cái này nó cũng giống như win-win đó anh, tại vì hàng năm á, nói chung là những cái sự kiện của Parkwell mà em đi mà em biết á, thì giống như là họ đều mời những cái bên họ giới thiệu công nghệ, nhưng họ mời những cái cơ sở nghiên cứu lớn trên thế giới đó là trình bày những cái mà họ đang nghiên cứu mà ứng dụng cái công nghệ của PacBio, thì nó là win-win thôi, đương nhiên là họ rất là muốn hỗ trợ rồi ạ. Dạ không biết anh còn câu hỏi, anh hài lòng với câu trả lời không ạ? Anh Linh ạ. Còn nếu mà khó quá thì lát nữa thì lại về mình hỏi thêm cũng được ạ.
Rồi, xin cảm ơn ạ. Yeah,
Dạ.
thank you.
He satisfied with your answer. Thank you. Can you please continue?
Okay. All right. I will continue. Let me close the chat box first. Okay.
Mọi người ơi, mọi người có câu hỏi gì không ạ?
Chị có thể đặt câu hỏi được không em ơi?
Dạ chị, cô Diệp hỏi trực tiếp tiếng Anh lên chị.
Hỏi trực tiếp tiếng Anh của mình hơi dở. So hi, nice to see you again.
Hi. Hello.
Yeah. Actually, very nice presentation. Thank you, and a lot of information. So I can see that every problem can solve by PacBio, right?
Thank you so much. Yes.
Yeah,
I hope so.
I actually fiber sick and SBB is really amazing. But actually, I saw in your presentation you don't have any comparison between PacBio and ONT. I saw it's just with Illumina, SBB with SBS, right?
Yep.
And for long read, you also compare with short read.
As well as
SBS
ONT as
is
well.
not with ONT. So do you have any research or result comparison between PacBio and ONT? I know that PacBio is perfect. I saw the PacBio result, but also now ONT they develop. There are many new version.
Yep.
And actually, they also have. I think they develop the super accuracy for base cooling. And actually, they develop the flow cell. So quite good.
Yeah.
Yeah, they develop super accuracy the bioinformatic
Yeah.
tool, and the flow cell also is now is improved a lot. So do you have any data the comparison between PacBio and ONT? So sometimes you said that PacBio give you the Q score 50, right? Even in the ctDNA, the liquid biopsy, and in the FFPE,
Correct.
you reach to Q50. But sometimes in ONT, I just read some information that they reach to Q20, 20 or 30 only, not 50. Cannot right now. But sometimes you don't need if because you have solution for any situation, right? For like liquid biopsy, cell-free DNA, and you have solution for the FFP. But sometimes it's just normal sample in blood sample. So now ONT also they develop for the whole genome sequencing for human. So do you have any suggestion or any comparison between PacBio and ONT result? Yeah.
I do have a lot, but I didn't put it here today, so I don't want to be like, keep bashing ONT, but I think the general statement from PacBio is always like, our accuracy is the best. We are the gold standard for long read sequencing, right? And like
Yeah.
what you mentioned, right? Like for hi-fi sequencing, our raw reads. So we didn't go through any base calling algorithm like AI. Our raw reads are already Q30.
Yeah.
Don't need any base calling algorithm or AI or machine learning to actually help to modify the bases to achieve only, I think, Q if I remember correctly, it was like Q28 or Q29. So 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 that's one point. Like, our raw reads are already Q30. We don't need to go through any, like special base calling algorithm that keep changing every 2-3 months. Then you have to reanalyze the results so that you will have better base calling itself. So that is one point. And I think if you do um if you do understand about I mean like base calling algorithm as well, right? So we know machine learning is like you will feed like a set of data into it, they will learn through it, and then they will, look through, and then they will, modify, I mean, improve your base calling so that, they have the higher accuracy, but still not as accurate as us. So a lot of time, this kind of base calling algorithm as well, it will overcorrect, right? Maybe some of the mutations that are lower frequency, they will treat it as an error and then correct it. So you wouldn't. So that's a risk as well, right? So we know cancer genomics, you are looking at low allele frequency, like some of these somatic variants, right? So they might be present in the samples in maybe 5% or lower than 5% itself. So if you're using this kind of base calling correction algorithm, they might treat it as an error and correct it, right? Then you won't be able to really discover whether is it a real somatic variants or not. So I think that is another point that you have to uh, look at as well. So, for our hi-fi sequencing, we don't do any base call correction. So every variance that we call, I think for clinical application, every variance matter, right? So that's why we feel that for PacBio, uh, we still a lot of these clinical applications or diagnostic labs, they will still use our sequencer for do this kind of applications lah.
Thank you for your answer. And I've had last question. You mentioned about the genome polish, about the SBB. You mentioned about the gene genome polish. So what do you mean in that situation? Gene genome polish, because you said that you don't use the base calling, right? Software. So now, hi-fi PacBio, you just use AI to collect.
No, 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 no,
Do
no.
you mean?
No. No, I was saying our competitor.
Yeah.
Yeah, so I mean we have our own base callers as well, lah, but our base callers are, just to call from our hi-fi sequencing itself. So there's no
Yeah.
correction algorithm in our base
Mm,
calling.
So what do you mean the genome polish? What do you mean in that case if you don't correct anything? So the meaning of polishment, the last I don't remember what's the number of slide, but you mentioned about the gene polish in here. Where is it? In some of your slide, you mentioned about the genome polishment. Yeah. So what do you mean? Polishment, it means that some in some problem with base calling. So you need to correct it, right?
Cô nói là cái polishing á hả cô Diệp, tức là
Polishing, đúng rồi.
she's talking about like
Polishing.
the polishing.
Yeah. Genome polishing. I saw in your SBB slide.
Oh, did I? Wait, let me. Oh, why does it matter?
Yeah, genome polishing, yeah. Here. So in your red box.
Yeah, I see it.
So what do you mean genome polishing?
I can get back to you, but if I understand, maybe it's, if you are doing like de novo assembly, like you want to make sure you have a complete genome itself, sometimes
Yeah.
it's short
Yeah.
to
Yeah, right.
some of the polishing, right? And then
Yeah.
I think maybe what they are trying to say, like, for this kind of repetitive regions, sometimes you need to use this kind of like highly accurate to actually polish it so that you have a complete genome itself, lah. But I can get back to you whether if
Yeah.
it's
So do you mean that just only SBB short read, right? You need genome polishing, but for hi-fi you don't need, because the accuracy is quite high. You reach to Q50.
I don't yeah, you are right. Like, for hi-fi sequencing, because we are long and highly accurate, you don't need to actually do genome polishing to actually, you don't need to polish it with short read lah, to actually, achieve, probably to do like de novo assembly or things like that. Yeah, but I think for the short read side also, why does it matter here? It means probably, with highly accurate short read sequencing, it can be used for genome polishing. Like, it will be able to enable better polishing. If let's say, you're using other long read platform, you need to do polishing, like with SVB, with this, like, because we can actually sequence through all this difficult to sequence region, right? And like this homopolymer region. Yeah, so it can be used for genome polishing. Yes.
Yeah. Okay, so please, if you have any literature or some information about that, so please send me via email.
Yeah.
Interested in that because it's very difficult for genome polishing. Yeah. So okay. So
Thanks.
thank you, Raylene. Very nice presentation. Thank you.
Nice to see you again. I think I will work with to provide you some of the literature.
Okay, thank you.
Thank you.
Cảm ơn các bạn, cảm ơn Thảo, cảm ơn.
Mọi người còn câu hỏi gì không ạ? À, mọi người ơi, I think no more questions for you, Mr. Ray. Thank you once again for your joining us today and supporting Mithila as always.
No worries. Thank you so much for your time.
Thank
So if
you.
you all do have any, I mean, if the participants do have any question or, you want to work together with PacBio for cancer genomics research, reach out to me or DKSH to I think Tue is in the call as well. Yeah, to discuss more. Yes. Thank you so much.
Thank you. Thank you.
Thank you so much, Ray. Thank you for the team. Yeah.
Cảm ơn chị Tệ. Dạ vậy chào nhau cái mình kết thúc buổi hôm nay ha. Chào mọi người bật mic chào anh Ray đi cho anh vui.
So have a good evening, Ray. How are you over there? So what time in your country now?
It's 9:50. Going.
It's not the same, the
Yeah.
same, 9:50. It's not too late for you?
Yeah, time to sleep.
Yeah.
Thank you for your presentation.
Thank you. Thank you so much.
Today
Thank
is
you so much.
today weekend, you don't go out?
For Dr. Lloyd.
Okay.
Have a good night.
VIP person, right?
Yeah.
Okay. So thank
Bye
you. Thank
everyone.
you.
Thanks a lot, Mr.
Bye.
Ray and chị Tuệ.
Thank you,
Bye bye.
everyone.
Chào mọi người
Thanks
ạ.
everybody.
Thank Ray.
This is
Bye.
Saturday. Thank you.
Cảm ơn mọi người.
Chị, bye bye chị.